Okay, guys, any questions before I get going with this stuff? I'm glad everything is working today. There were, technical difficulties. Sometimes it's just as simple as having something plugged in. This little guy that I never use, apparently everything runs through that. And it had got unplugged for one reason or another. And so that was why nothing would communicate. The computer would turn on, but that was the issue. So it took multiple people in here before they were like, oh, this thing's not even plugged in. That's good stuff. So we are functioning as we should be, as I have hoped, and failed, except when I was teaching from my house. I have been unable to get all of this stuff to record at the same time as we are doing stuff, but it is functioning now. So this will be posted later if you are interested and want to review. And for those of you that aren't here, there you go. If you've got questions or concerns, if you are not present, shoot me an email and I can go over stuff with you if you need. So we've got a few chapters that we need to get through before the exam. The exam is a couple of weeks down the road. Part of me felt like it was supposed to happen Thursday. It was supposed to open up, but we're covering a couple more chapters than just chapter 3. So we've got 3, 4, and 5 that we're going to cover this week and next week, and there should be no issues getting through that stuff. So if you guys don't have any questions for me, we will start thinking about some cells. We'll start thinking about some details about all sorts of different types of life, and not all cells are the same. All right, we will think about bacteria, what we call prokaryotes, and then we'll think about pretty much the rest of everything else. They're eukaryotes. All right. Technically, deep down, there's one other group called the archaea. They are also prokaryotes. They are sort of like bacteria. There are a few deviations with those guys that make them a little bit different. We'll talk about them too. But really, we think about the microbes like bacteria and then other stuff. And we want to make some comparisons, compare and contrast some of the variation with these guys. And that's really the focus of this chapter. The next 2 that we will discuss before the exam have to do really with energy. And we'll think about cellular respiration in chapter 4. And then chapter 5 talks about photosynthesis. Where do we get the sugars in the first place? All right. And it all really gets tied together. And as we start describing some of the stuff in this chapter, we will go through more details in the next 2 as we start talking about, well, this is what happens in the mitochondria or in the chloroplast, respectively. All right. So there are all sorts of details when we think about cells and we try to visualize some of these things. I know you guys will have an opportunity to look through some microscopes and see some stuff in the lab, some plant cells. Those are onions, and that's what you will look at. You'll look at some other stuff as well. Just to get kind of some perspectives, right, with that. So we've got some nasal sinus cells. This is all light microscope, the first 2, and our last one is with a bigger microscope, an electron microscope that allows us to zoom in considerably more. With the regular microscope, what you guys are going to work with in a lab, what we think about using here, I mean, we technically have one of these, but it's not for just anybody to roll in there and be like, oh, I want to use this. I know we've got one, but I've never got to use it. All right. So The first two allow for magnification and depends on the types of cells that we are zooming in on. But a regular light microscope can give us about 1,000 times magnification. That's not how magnified we are on these cells because they're bigger. They're eukaryotic cells. They're already pretty large, and zooming in that much is going to focus in on details of an individual one pretty much, not showing you multiple ones. The electron microscope allows us to zoom in considerably more, thousands of times, upwards of 20,000 or better times magnification compared to the 1,000, right? And you can see here the ruler bars trying to put stuff into size perspective. 10 microns is very small. Here we're bumping it up. This is 50 microns. And over here, 500 nanometers, right? Considerably smaller than what we're talking about over there, all right? An order of magnitude, all right? And with that, that puts it into a little more perspective, all right? This guy, so we're looking at animal or our cells, plant cells, and here we've got a bacteria, a couple bacteria. And these guys, Vibrio, so what is it? Tasmaniensis Vibrio cholerae causes cholera. This guy, I can't tell you what it does. Probably nothing. Maybe lives in a Tasmanian devil. Okay, so these are a couple of different light microscopes. It depends on the circumstances as far as what you might see in a lab. This is a compound microscope with multiple objective lenses that are going to allow us to see the nasal cells or the onion root tip stuff that we were just looking at, right? So also looking at stuff like bacteria, not the electron micrograph that we just saw, but you could see Vibrio under the microscope and they'd be very, very small if you compared it to the nasal cell, right? Much smaller. So these guys allow us to see stuff like that. This next guy right here, similar, but it's a little bit different. As you look at it, there are some variations with this, with these two. This microscope is a dissecting microscope. So it's something for larger samples, maybe a full organism, right? It really kind of depends. And you can see there's multiple eyepieces and potentials for maybe someone else to be looking in, maybe the instructor peeking in there as well, just to make sure that what you think you see is actually what you should be seeing, right? Oftentimes, like in microbiology labs, the first real experiences of working with microscopes and you stain some stuff up, start looking at things, almost always students are like, oh, I have found stuff. We always want to look in there, not just because we're microbiology nerds and we want to see everything through the microscope. We want to look in there and make sure that what you're looking at is really what you should be seeing. And almost always the first stuff you think you see is dust. It's just little speckles of nothing, all right? Not really, I mean, dust is something, but it's not really what you want. Or it's just really out of focus and you're seeing well, little blobs, but it's really the glass from the slide or from one of the lenses, and you're not focused as you truly should be, all right? So that's why we're seeing some extra eyepieces on there, so maybe someone else is checking stuff out. There are other microscopes where cameras are attached that can allow things to be projected, where you can see some of that stuff maybe on a monitor. So there's a lot of different details. I'm not going to go through all of the stuff, but knobs that allow you to focus. Basically, it is going to manipulate, in this case, the stage, bringing it closer or farther away. A space for the slide to go on, and then your light source. Here, the stage is stationary. It's just the base of the microscope. And what moves is the microscope itself. As you focus up and down, it brings it closer or farther away. Dissecting scopes don't give you near the magnification that you get with a compound scope, but we're not using it for that same purpose, right? There is really some pretty cool stuff that, you know, either of them you can see. I typically Most of my stuff is working with a light microscope, seeing bacteria and stuff. That's typically what happens, all right? Occasionally, those things, all right? And I was dealing with a class, dealing with a lab, this stuff, and we were working with some of these things, and that's when I really started to just kind of be like, oh, well, what if we looked at this or that? And I'm like, oh, my kids need to see this, right? There is all sorts of crazy things that you don't think about. Slapping a dollar bill under a dissecting microscope and just looking at some stuff is crazy. There are little spiders crawling around on the dollar bill. There's all sorts of stuff on there that you don't even think about. A penny under the microscope, the older pennies when it's got the Lincoln Memorial on the back. Abraham Lincoln is actually sitting in the Lincoln Memorial if you look at it through a dissecting scope. When you just look at it with the naked eye, it just looks like the Lincoln Memorial, but you zoom in on it and sure enough, there he is, right? That's pretty stinking cool stuff, all right? And there's all sorts of other things, all right? Pick a flower and slap it on there, and then all of a sudden you're like, holy crap, there's little bugs everywhere on this, right? And you don't see it, right? That's pretty cool, all right? So there's all sorts of different things, depends on what you're trying to look at, right? And ultimately providing some opportunities to examine things in a little more detail, all right? There are definitely, if you are interested in just seeing some of that stuff, numerous Google images that you can see this type of stuff, or you flip it over to like electron microscope and see like dust in your house. You'll never think of it the same way when you actually see what that stuff really looks like and some of the organisms that are living within it. You're like, holy cow, I must take a shower. Doesn't matter, you'll never get it off of you, right? So here's a couple of different bacteria, Salmonella, these guys, all of the little purple speckles, right? And this is something that, you know, I think pretty unimpressive, all right, to see this stuff. Just little specks. It's all just kind of glued onto the slide, if you will. It's not moving around. It's not alive anymore, right? And it's very, very small, right? I look at this image. It's not the exact same image, but it's the same organism. It's both Salmonella. And I look at this and I'm just like, holy cow, that's awesome, right? That stuff is cool. And that is zoomed in with an electron microscope. You can see the ruler bars with this. Here, this is a micron, right? And over here, this is 25 microns, right? So we have zoomed in considerably, thousands of times to see this Salmonella as a humongous pink monster compared to the little purple dots that you're seeing there, all right? So it just depends on the tools that you might be using, all right? And like I said, as we were kind of starting this stuff, these bacteria are what we call prokaryotes. They are very simple compared to our own cells, all right? But just because they seem super small and simple, they still carry out the same sorts of stuff that our own cells are doing as well. They're growing, they're replicating, they're pulling in nutrients basically all of the time, pulling in nutrients and metabolizing, stuff that we'll discuss in the next chapter with respiration. They're doing this stuff too. They've got similar things that they have to carry out, things that we would say, well, that's essential for life. You've got to pull stuff in. You've got to metabolize. You've got to replicate. All of these things, it doesn't matter if you're talking a human cell or a bacterial cell, they're doing similar things. When we get to larger organisms and start thinking eukaryotic cells, well, now those roles are partitioned, different functions with different organelles within the cells, but it's still doing similar stuff, right? It's just saying, oh, you do this over here, you do that over there, we'll all use it and work together, right? And the little bacteria does it all on its own, right? And we'll show some details with that as we progress through here a little bit more. All right. So here we're looking at a couple of cells, normal and abnormal, normal and abnormal. So these are cervix cells. So a pap smear and basically evaluating this smear under the microscope to see these cells. If they look normal or abnormal, will clue us into some potential issues, right? So we've got, as I said here, normal cells, and there are a handful of cells together, even though it looks like kind of a big blue blob. You can see separated out a handful of lines, and you can also see the nuclei indicating all separate cells. Eukaryotic cells have the nucleus. And when you look at them stained out, when you look at a stained cell, eukaryotic cell through the microscope, that's almost always the first thing that you see is a big, dark, maybe purple, maybe almost black nucleus that you see in the cell, right? And there should just be one of them, all right? And it's usually so large that it sticks out like a sore thumb, like a bullseye. All of a sudden, you're like, there it is. That's the nucleus, right? These cells over here are very different, right? They're much larger, and the nuclei are humongous, right? And what happens in circumstances like this with these cancerous cells, you get fusions of cells. So now what should have been a handful of cells, maybe half a dozen cells, now there's maybe 2 or 3 of them, 1 or 2 gigantic cells. And the nuclei also, there's fusions within that. These things become, well, this is cancerous cells. So this is infected with papillomavirus, HPV, which fortunately now we have vaccines against some of that stuff, not all of it, but some, to prevent this. And this ultimately sets the stage for cervical cancer. As we see these cells that get basically messed up as they fuse together, they no longer function like these normal cells should be functioning, right? They replicate kind of uncontrollably, coming in contact with others and causing multiple fusions, issues for sure. Okay, so we want to think now some details, some comparisons and some differences between some of these cells. So this is a generalized structure, all right? There's all sorts of variation when we think about prokaryotes. Some of them are this shape. We call these guys rods or bacilli-shaped bacteria, kind of elongated. There are others that are nice little spheres. We call those guys coccus, single one, or many. We call them cocci. So rods or cocci are the typical shapes. Some of them are kind of corkscrew shaped, spirillum or spirochetes. Doesn't really matter, all right? With that said, this generalization is trying to show us, here's what we think, all right? There's all sorts of potentials. Some of them have all of this stuff, and some other bacteria may not have all of it, all right? It kind of just depends on who we're talking about, all right? So the center, the guts of it, if you will, would be the same. There's a chromosome. It's a circular chromosome, like a gigantic rubber band kind of on the inside of the cell. They say it's localized in a region called the nucleoid. There's no nucleus, right? And it's really kind of depends on the cell and its process of living as far as where we might see it. If it's in the process of replicating, there'll be some more stuff happening on the inside than if it's just hanging out with it, all right? So chromosome, one chromosome, circular, all right? The little speckles in there are ribosomes. Ribosomes are important for protein production or protein synthesis. They're like the workbenches where the messenger RNA gets transcribed, or translated, excuse me. So we'll see some of that later on, right? There's also the light blue stuff, the fluid that this stuff is suspended in. The cytosol, right, is basically that stuff, although I don't think we even have that listed in there, but fluid, all right? It's not just empty space. These guys have a cell membrane. The darker green bit around the inside is the cell membrane or plasma membrane. And there are definitely some similarities, a few differences comparing our membranes to these. But there are some similarities. The phospholipid bilayer is pretty much the same. Some of the other molecules might be a little bit different, right? So cell membrane. After the cell membrane is a cell wall, this darker bit, third one in there, all right, is a cell wall. Almost all bacteria have cell walls, right? There's one called mycoplasma that's kind of an oddball, and it doesn't, and it will take on any sorts of shape. That's for microbiology class, not for you guys, all right? But there are some differences with the cell walls, and some of our bacteria have super thick cell walls, and others very thin, like a almost, you know, it's like a big thick down comforter versus a sheet, right? Very different as far as the coverage might be if we think about it like that, right? So those bacteria with super thick one, we call those guys gram positives. They've got a very thick cell wall. The gram negatives, very thin, right? And that becomes important when we think about maybe trying to treat infections. Different types of antibiotics work differently, targeting one or the other, usually. All right. Sometimes they prescribe a drug that's called a broad spectrum. Those types of antibiotics are effective against most. So it's kind of a kill them all approach. And deep down, if you're taking antibiotics, you don't want to take those, right? Sometimes that's what you're prescribed. But if you had a choice, you're better off with specifically targeting this type of bacteria, not kill them all, right? Because it sets the stage for other issues, all right? We'll talk a little of that stuff later on with some problems that we might see, all right? So cell walls, the thickness of that we can use to differentiate these bacteria and kind of set them up into different groups, all right? And variation with that. The outer bit here, this lighter green stuff, this is a sometimes, right? Some bacteria have it. It's called a capsule layer. It's like a sugar coating, right? Some of them have it, some of them don't, right? So with that said, if we're thinking about bacteria that maybe infect us and cause disease, if they have this capsule layer, it gives them an advantage, right? Now, all of the sudden, what we should be seeing as a bad guy, as a foreign invader, our white blood cells just see sugar. And they're like, oh, this stuff should be there. Sugar's okay. We like sugar, all right? And so it's like a little cloaking device. That bacteria is hidden under its sugar shell that allows it to go undetected. And that potentially allows it to establish itself and cause disease because it doesn't get seen by our immune cells, right? There's other stuff that helps them to stick around, like maybe plaque on your teeth, lots of capsule layer on the bacteria, sticky, that allows them to adhere to surfaces. And that also sets the stage for potentially causing disease, right? Some of our bacteria have these big flagella, whip-like appendages that allow them to propel themselves through their environment. And they may have a single one, they may have multiple, it might be all over their entire cell, right? We can use that a little bit to differentiate. We don't care about that here, all right? So it helps them move, all right? And it's only those that are broad-shaped that have flagella on them. Some of them have pili. The pili are little projections that help, also kind of like the sugar coating that helps those cells to kind of stick around. The pili help for attachment so they can stick to stuff. It also might help for one cell to communicate or come in contact with another bacterial cell, and they can communicate through pili, right? Exchanging genetic material potentially, all right? So pretty simple, pretty bare bones. And as we look at the inside, the guts of these guys, there are no membrane-bound organelles. There's no vesicles. All we see, nucleus and ribosomes, right? So there are definitely some differences with this, right? Where we're not inside of a nucleus. Transcription and translation happen virtually simultaneously. That doesn't happen in eukaryotic cells, right? We've got to do that in steps, right? We got to ship it out of the nucleus in order to be translated, right? There is no nucleus. That all happens at the same time. There's other stuff in eukaryotic DNA that bacterial DNA doesn't have. And so we don't need any sort of modifications. We just immediately start reading it, all right? Definitely some differences with that, all right? We'll talk metabolism and some of that other stuff later. But our bacterial cells and our own cells, we're doing similar stuff. And how it happens, we'll go through that a little bit later. I'm not going to go into it right now. So this gives us some perspectives. Almost always, when we start talking cells and we start thinking microscopy and stuff like that, we always show something that kind of tries, we sort of hope, tries to put it into perspective, all right? So we can see, we'll move over here to us, obviously visible with the naked eye. We could see ourselves. We could see some cells with the naked eye, like an ostrich or a chicken egg. You probably have some eggs in your fridge, probably not ostrich eggs. Probably not, but you never know, right? We could see all of this with the naked eye. Frog eggs, a human egg, maybe, barely, all right? Probably not, all right? Write it about this. That's where we kind of cut it off. And then the light microscope that we first started showing, all right? This is where we start thinking about visualizing some of this bacterial cells often, all right? The plants and animal cells, as we see here, a lot more going on. We're going to get to these guys here in just a second, all right? Plant and animal cells with lots of business on the inside, as we showed our bacteria just the last slide. There isn't that stuff going on the inside, all right? It's different. The mitochondria that we see in plant and animal cells that also kind of looks sort of bacterial-like. We'll hit that in a minute. Moving from this point, that's the light microscope. Now we see the electron microscope right here, where we might visualize some plant and animal cells as well. But we also get smaller. We showed you the salmonella that was gigantic with the electron microscope, but you can also see proteins and lipids. You can see other substances, and it depends on the types of microscopes. Some scan the surface, and some transmission electron micrographs go through, giving us more details like maybe internal structure of those cells. And it shows down here, down to the atomic level, technically with a regular electron microscope, you're not seeing individual atoms, right? I believe with an electron, we can get down and show hydrogen molecule, two hydrogen atoms linked together. You can visualize that, but an individual hydrogen, not, right? That's just a little bit too small, but I think there is another atomic force microscope that allows us to get a little bit deeper into it. So with that, this basically tries to give us, and both of, there's so much stuff on there, that's pretty busy. Trying to make some comparisons between plants and animal cells, or animal and plant cells, as we see. If we look closely at this, there are definitely things that both of them have, right? There is a lot of similarity here. There are a couple of things that we see different between them, right? Depends on the type of cell, right? So real quickly, we'll go through this. We'll see more details as we progress along, but quickly we can see a few things like the nucleus, all right? Big, usually purple or dark colored, all right? We could see that in both of them. We can see the mitochondria, these little kind of bean-shaped guys, in both, right? There are some other structures that we also see, like the Golgi, the smooth and rough endoplasmic reticula, these guys shared, right? Some lysosomes in there, the chloroplast, the little green bean looking guys, we see those in plant cells, but not in animal cells, right? And that's where photosynthesis is going to be occurring, all right? The mitochondria is where cellular respiration happens, and both plant and animal cells have that. They both go through cellular respiration and metabolize stuff like sugars, right? So there's other stuff like some of the microfilaments, cytoskeleton proteins in an animal cell serve as like an internal scaffolding to provide shape to those cells. Some of the microtubules in an animal cell are important when we think about cellular reproduction. So separating the chromosomes to opposite poles of the cell, these microtubules help to do that. We don't see that stuff in a plant cell. They don't have that. They separate and then they divide by planes of division dependent upon where it's at. So those root tips keep growing in an onion that we showed an image of a second ago. They're growing down and we see the division happening that way. There's also a little bit of the other division, but it's not just wherever. It's constrained by specific factors. The same idea with our own cells and recognition, one cell touching another or whatever allows it to know where it should be or not dividing. We're talking cancerous cells, that's different. And those cells don't care. They don't care if they're touching another cell, they'll still divide, right? But normal cells don't do that, right? They're constrained by what we call contact inhibition. I'm touching something, I can't grow that way, right? So that's all dealing with different proteins or receptors on the surfaces of these cells. We're not talking about that stuff in here right now. So there's other details, but some similarity as far as the nucleus and the structures within it. It's the same sort of idea between the two. So this is an image of the membrane, the plasma membrane or cell membrane. And we really are looking at like eukaryotic cells. There are some, like I said, similarities when we're talking bacteria, but there's a few things that are slightly different, just kind of depends on the bacteria. So this is again, generalized image. The little red guys with the brown tails, if you will, are the phospholipids. And these phospholipids form a bilayer. So it's two together. The tails, these little brown guys, are hydrocarbon tails. And we talked about this stuff Thursday. These things are these tails, this lipid, doesn't like water, right? And so you get one tail associated with another tail forming this bilayer. And this bit right here, the inside, hates water. That's super important when we think about a cell. It helps to keep the inside, which is water, and the outside, which is water, separate, all right? You want the stuff in, and it's more than just water on either side, but it's liquid, all right? You want to keep what's inside in and what's outside out for the most part, all right? There are definitely things that you would like to allow to pass one way or the other, all right? But you don't want stuff to just move whenever it feels like it, right? So this hydrophobic region, this water-hating region on the inside of this membrane provides a semi-permeable barrier. It lets some stuff through, all right? Most stuff can't just move right through, right? So the red bits These are the phosphate groups. It's a phospholipid bilayer, right? And the red bits are hydrophilic. They like water. So they associate making the inner and the outer bits of that membrane that comes in contact with water. And the tails keep it apart, right? We can slice through this membrane and unzip it, if you will, and pull these guys apart and then potentially see some of these blue guys or other stuff. Does it go all the way through? Is it on the top? Is it on the bottom? It depends. Some of these proteins, the blue guys, as far as their roles inside of the cell, right? On the surface, this would be the surface pointing to the outside, right? On the surface, we can see some of these other structures, the blue guys, glycoproteins, so sugar protein mixture that serves as like a molecular fingerprint, letting our cell know this is our own cell. Other things, the green guys are glycolipids. So sugar lipid combo, and it's a similar sort of an idea as a recognition, as a communication from one cell to the next. They set the stage for that, right? As I said, these blue things, these are other types of proteins, and they may form channels, like little holes in the cell that allows things either in or out, or maybe both. There are these things aren't just like a hole that lets anything through. They're specific, allowing one type of a molecule in or out, right? There's other things that act almost like switches, coming in contact with something, stimulating a reaction to occur inside of, or maybe something to happen on the outside of the cell, maybe allowing as a receptor something to come in and link up to it. There are all sorts of potentials as far as what these proteins might be doing. Some of the yellow bits on the inside of this membrane, these are cholesterol molecules, a type of lipid. And they're important inside of the membrane because it lends to the fluidity. It lends to the ability of these molecules or these membranes to kind of move, to take on various types of shapes. It's impacted by temperatures. Lipids, as it gets cold, tends to slow down and not be as able to move. Things that are warmer makes it more flexible, right? So there are definitely some issues with some of that stuff if we're thinking about working with stuff on the bench, not in our own bodies, right? Because that should be warm to begin with, right? And there's differences between types of cells as far as the amounts of cholesterol that you might see in the membrane, but it is an important structural component of that. Below all of the spaghetti looking stuff, these are cytoskeletal proteins. So these are protein strands that, like I said a second ago, help to give it sort of an internal shape. It also helps with all of the organelles, all of the different structures that we see inside of these cells, helping to keep them tethered or hold them in place. It allows some things to kind of move and like almost like a train track or the interstate where things can move right on these filaments, right? So there's all sorts of different potentials with that, dependent upon, well, the type of cell and what might be happening, right? You should expect that maybe the stuff we see in a skin cell might be different than the stuff you would see in a liver cell or in a whatever kidney cell, all right? There's going to be variation, but they still have membranes. They still have this stuff. It's just the functions of what they're doing and the inner guts, if you will. There might be some variation with that, right? Liver cells have lots of smooth endoplasmic reticula within them because it helps to detoxify stuff. There's also lipid synthesis that we can see occurring in there as well, but mainly like detoxifying, all right? So it just depends. Here we can see some of these proteins, all right? These structures, the microfilaments, intermediate, and microtubules. Three types of proteins that provide some structural characteristics, and these microtubules are important, like I said, in an animal cell for cellular replication. You don't see this stuff in plants, right? Some of the other stuff, but not these microtubules, right? So microfilament, intermediate filaments, this is like the internal cytoskeleton tethering and anchoring stuff. And this was cellular replication. The microtubules link up to chromosomes, and microfilaments helping with the cell as it replicates and goes through cytokinesis. It will pinch and constrict and form two new distinct cells, right? But they're all types of protein with different specific roles with that. So we saw a little of this when we looked at the animal and plant cells, and we're basically just blowing it up. That's why I didn't go through great details as we looked at that slide in the first place, because I know we got bigger ones to kind of expand upon as we think about it a little bit more, right? So here we're looking at, well, the nucleus is the big part of what we're looking at, but it's more than just that. Right, it's part of what we really call the endomembrane system, the internal membrane. We're not talking about the plasma membrane that's the outer boundary of our eukaryotic cell, right? We're talking about inner stuff, right? And there's all sorts of details and ideas about this stuff. One quick thing I mentioned, like partitioning jobs, doing different things, right? We separate stuff out in a eukaryotic cell, and it makes it more efficient, right? Why do this all throughout? We'll do it right here, and we will use those products or whatever it might be, right? So, a little bit of stuff about this, all right? The eukaryotes, like what we're seeing, the part of that here, eukaryotes compared to prokaryotes are much bigger, right? Almost 10 times bigger than a little bacterial cell. There's a lot more space. It's still small, but there's a lot more space. There's more potential for things to be going on within them, right? But you can't really get much bigger than that. I mean, the ostrich egg is pretty stinking big, but when we're thinking about like our own cells within our body, they're small, right? They're constrained by, specific biochemical and physical constraints that say you can't get much bigger, right? And part of that has to do with how stuff is going to move in that cell. And we think, most of these things move by diffusion. So an area where it's really concentrated to an area where it's not so concentrated. And it doesn't take any energy for that stuff to move, right? If your cell's nice and small, that's pretty easy for that stuff to happen. And it's pretty easy for nutrients to get into the cell and to be tapped into and utilized, right? But if you have one cell that is gigantic and you need to get nutrients from one side to the other, that could take a tremendous amount of time, right? So we're constrained, we're small, whether we're talking bacteria or eukaryotes, right? We're small. So if we think about it like ourselves, right? Maybe it's just you. You have a little apartment, it's all fine and good. And then maybe you find a significant other. You get a sweetheart, then you decide, you know what? We ought to live together, right? So you move in together, and maybe it's all fine and good. Maybe you've got ample space. Maybe you decide it's a little cramped, right? Now maybe all of a sudden, love is in the air. Maybe there's a little one that's ready to hatch out, right? We could potentially up and move. We could go someplace else. We could get a bigger place, right? Maybe we own this spot and we got money that we don't even know what to do with. So we just add on, right? We could build a new little nursery for this little bundle of joy that's going to cost me a fortune, right? We could do this, right? And we could expand. We could do all of these things and get more space, right? But our cells, they don't really do that, right? And if we shift from a little bacteria hanging out, minding its own business, and we start thinking about bumping it up to eukaryotic cells, all of the sudden, the potential to do more is there. There's more stuff happening. There's more nutrients we're pulling in. Now, all of a sudden, we're thinking, geez, what could we do? And instead of expanding, because you can't get so stinking huge that it becomes prohibitive, right? We expand in. That's all of this stuff. Pushing membrane to the inside, this endomembrane, internal membrane, all right? Now we've added on the inside, and we're still in the same space, but all of this extra membrane has increased the surface area tremendously. Now you've got massive amounts of stuff, but it's on the inside. It's not making my cell, you know, huge, right? But it is on the inside, right? And with that, we see organelles forming and different roles, different partitioning. The first bit, walling off the nuclear material. DNA is very sensitive to chemical shifts and changes, and altered pH could destroy it. Now that's the end of that cell. But if we protect it in a lipid membrane, now it's encased and it can't be touched by biochemical reactions that might destroy it, all right? We've protected it, all right? So this membrane, this outer bit of the nuclear membrane, comes right in contact with this next bit of the membrane. They form a continuum, right? And the endoplasmic reticula, the rough bit, are all of the red dots, all right? The smooth we will see in another image. So we've got the potential to basically partition this stuff, keeping the nuclear material inside and then allowing it to be released through these nuclear pores, little doors or channels that lets messenger RNA out. It's also going to let some stuff in, but that's a different story, all right? So the material comes out, and almost always it associates right with the endoplasmic reticula that's studded right next to it. And we start building protein, right? There are some free-floating ribosomes, and that's for different stuff that's happening inside of the cell. We keep those proteins right there. Other stuff might be manipulated and moved and transported, and it has to go through the rough endoplasmic reticula first. All of this stuff is an invagination, a pushing or an addition to the inside, making space for more things to happen all in the same surface area, all right, of that cell, all right? So membrane layer, all right? And the red bits on here are ribosomes. This is where protein synthesis occurs, right? There are others, the smooth endoplasmic reticula with no red bits on them, all right? And we see those pretty close to this same area, all right? All still functioning together. The Golgi apparatus that we see here would also be very close to this, all right? And this is an electron micrograph showing this. We can see Golgi all stacked up together, and then we've got two different sides, right? Cis and trans, right? So with that, right, Golgi, I always say, you know, Golgi, it's like the post office, but it's a little easier to understand what Golgi is now if I say this is Amazon, right? Golgi is the Amazon distribution center because Amazon has all sorts of stuff, right? Pretty much whatever you want, right? You could get that crap on Amazon, right? And maybe it just so happens that there is one distribution facility and it's got all of this crap in there, all right? They know where it's at, all right? They know where it all is, all right? And maybe you're buying a bunch of stuff and you're thinking, I want to save the environment, send it all in one box. I'll wait an extra day. That's all fine and good, all right? That's what Golgi does. It's the same sort of an idea, all right? It's the collection and distribution center, and it knows this crap should be packaged with that crap. We should put it all into one box and we should ship it out, right? And that is what Golgi does, right? It collects through the receiving and ships it out through the transend, right? Shipping and, or yeah, receiving and shipping, right? We get it, we collect it, we package it, and export it. So proteins can be synthesized in the rough endoplasmic reticula. They are in a vesicle, which we can see all of these little vesicles in here, right? Harboring materials. They fuse with Golgi. They dump their contents to the inside. And now we collect some of these things together, all right? It might be multiple types of proteins. There could be some lipids associated with that stuff. So there's other vesicles coming from the smooth endoplasmic reticula. We link all of this stuff up together, packaging it based off of whatever it is we're collecting, right? And shipping it out in another little vesicle. This might become a lysosome that's full of all sorts of digestive enzymes. We'll see a picture of it in just a second. It might be other proteins that are getting exported to the outside of the cell, maybe staying on the surface or maybe being released out, period, right? It depends on what's happening, right? But Golgi ultimately is what collects and ships substances within the cell, right? And it's another part of the endomembrane system, more membrane that is pushed to the inside to do different jobs, different roles, all right? So dependent upon, you know, smooth or rough, moving to Golgi and then shipping them out, all right? So this image is a white blood cell, a macrophage. These guys are some of our body's first responders when we think about disease, all right? And they're, you know, typically thinking about stuff like in the tissues, your skin, or other areas in your lungs, there's some macrophages. And if something like a bacteria happens to be ingested by these guys, they're going to destroy them, right? So the lysosome, I mentioned it just a second ago, maybe we've made all sorts of things, proteins that are going to be destructive. We collect all that together in this one little vesicle. It's full of all sorts of stuff, right? So the bacteria gets ingested by the macrophage and it eats it by a process we call phagocytosis. It's the cellular eating, right? And in this case, cellular killing, because we're eating something alive, right? But some of our other cells can pull stuff in too, and it's like bigger molecules that just can't move through the membrane. So this little brown guy's a bacteria, it gets ingested by this macrophage, we pull it in, and as it comes into the cell, it's in a vesicle. It's a membrane-bound vesicle, part of the plasma membrane. It pinches it off, right? So now we've got a bacterium encased in some vesicle. This vesicle is going to fuse with a lysosome, giving us a secondary food vacuole, if we want to call it that, or a phagoolysosome is more scientific. A phagoosome is this, where it's just been phagocytized, and then with the lysosome. All of the proteins, all of those enzymes, the substances that are going to destroy stuff. It's almost like you're dumping a bunch of acid on this guy, right? That could be an example. It's going to blow it apart, all right? Once all of the guts of this hit that, it breaks it into little bits, right? Destroying it and making it so it's no longer an issue, right? There's technically some other details that our little macrophage might do, right? This is one of the main things, and we think about just getting rid of these bad guys, right? But macrophages are part of our immune response, and sometimes these guys will take little bits that are often protein, right? Little bits of that bacterium or other foreign whatever, and it says, oh, this is like a fingerprint, right? This is like, this says this is this bacteria, and it's bad news. I'm going to take that little piece, and I'm going to put it on the outside of my surface. So through a vesicle, it pushes it to the outside, and now you've got this substance all on the outside of the cell, and it shows it to other cells and says, look at what I just ate. It's bad news. And those other cells do stuff like make antibodies and provide some other lines of protection so that if the antibody happens to see those things, it initiates other responses as well, all right? And the macrophage, so it starts this whole process by eating something, but then it does like some show and tell. And it says, oh, like I said, look what I just ate. Here's little bits of it. You should go try to find them, right? So it's super important in recognizing, but also stimulating an immune response based off of what it ate, right? What it came in and found, right? So you hope that it's all bad guys, right? But sometimes that's not what happens. And we think about stuff like allergies. You get, different types of allergies. And it's kind of a similar idea like this. Something gets brought in and it really isn't or shouldn't be bad news, but it gets treated and presented as if it was bad news, and we mount a response against it. And now you've got an allergy against whatever it is, right? And I'm willing to wager half of you guys in here will raise your hands and say you have allergies, right? Just depends. Could be hay fever. Usually stuff springtime is when it gets you, but just sort of depends on the circumstances. Shellfish and peanut and other food allergies are other possibilities as well, but hay fever is pretty stinking common, and most people would say, I have that, right? So it all kind of just depends on how that response was stimulated in the first place. And for allergies, it's not really being stimulated the right way, right? Okay, so this is more of what we've just kind of described. So the nucleus, the rough endoplasmic reticula, over here is the smooth endoplasmic reticula, and then the Golgi apparatus. All of these guys all kind of very close to one another because the jobs they're doing needs to be almost kind of like an assembly line, all sort of right together, all right? So again, in the nucleus, make a messenger RNA saying, I want to build this protein. We leave from the nucleus, we start associating with the rough endoplasmic reticulum where we do make proteins. As we're making all of that, it's moving through and we export that out. So here's this little protein for export. It comes out in a vesicle from the rough ER, fusing to the cis side, the receiving end of Golgi. There's other lipids, other substances. We're not showing any of that, but it can be coming from the smooth, also fusing. And with that, we move through, package, process, putting all of these things in the same box, if you will. And then in this case, the substances inside of here are going to get exported to the outside. So this vesicle fuses to the plasma membrane and releases it out, right? As we look at this, and we can see from Golgi to the membrane, we can see another image with that macrophage where some of that membrane goes away, right? The membrane is a fluid model, right? We can add to it, we can take away from it, and overall, the general shape stays pretty much the same, right? Because it's constantly kind of changing. It's in a state of motion. Where we're able to give and take a little bit. So we're giving some, right? And put some more membrane, this guy, right back into it. But we can flip it back to the other image where we've lost some of it, right? It doesn't really matter, right? As we move through there. If we were constantly adding or constantly removing, well, then that ultimately sets the stage for issues, right? But that's not what happens inside of a cell. Do you guys have any questions? I realize that I'm like, I just started talking and I don't even know if I took a breath. I think I took 1 second and had a sip of my tea. All right, but I need to give you a second to kind of think about it. All right, this image, also an electron micrograph. We've zoomed way in on it. If we were trying to see this through just a regular microscope, it's not going to be too impressive, right? So mitochondria, and we showed that little cartoon image a second ago. The mitochondria and the bacteria were pretty similar size-wise, right, as we looked at, right? So through a regular microscope, they're not going to be very impressive, right? Here, a micron, all right, and so this looks like it's maybe 2.5 or 3 microns long, maybe about a micron for the width, all right, which is about a bacterial size, right? And we'll touch on that in a second, right? So this mitochondria is, it's kind of like the energy source within our cells, right? That's kind of what the mitochondria is like. It's really like the charging station. If you've got a bunch of rechargeable batteries, the mitochondria is what's going to recharge all of them, right? And it's something that is super proficient at charging, right? And the little batteries, the ATP molecules that are the batteries of our cell, the energy that we need, are constantly being used and recharged. And it's the mitochondria that's doing this stuff, recharging, right? And it does it by the foodstuffs that we consume. We take in energy in the foods that we eat, and we harness that energy into chemical energy that our cells can use. And we'll touch that on that stuff in the next chapter. And the bulk of it's happening inside of the mitochondria, all right? So there's a handful of things, and we will discuss more of it in the next chapter. But the mitochondria is, sometimes we describe it as like a bag within a bag. It's a double membrane bound structure, right? An outer and an inner membrane, and there's different things happening within this organelle, right? So the outer membrane forms this margin around it, right? And really it's, I don't know what we got going on there. The inner membrane is, so outer and inner, right? And we can see some infoldings with that, invaginations with that inner membrane that go kind of throughout, providing some space, all right? These cristae are these inner bits of the membrane that are kind of poking through, almost kind of convolutions like your brain, as it's all, you know, shaped like that. That's sort of what's happening in there, increasing the surface area on the inside, all right? And the matrix is the space on the inside there, right? The mitochondrial matrix. So there are important things happening on this inner membrane, right? Where there's numerous enzymes, proteins that catalyze reactions, basically harnessing energy is really what we're thinking about, right? And in this space, in this matrix, is where this stuff is occurring, these reactions, and they interact with proteins on that inner membrane, right? We'll see more stuff and talk about the inner and the outer membrane and the space in between and what we do with that in a bit, right? The next, so chapter 4, that stuff we just looked at, cellular respiration, details about mitochondria, what sorts of things are happening. And then chapter 5, with photosynthesis, speaks about this structure more than other stuff. There's still mitochondria and other things happening. But with photosynthesis, we think about another type of organelle that's also very bacterial-like, as we look at some of these details, right? A little bit later we will. And there's multiple kind of double membrane ideas as we move through here, right? Kind of like the mitochondria, but a little bit different, all right? So there is an outer and an inner membrane. There is an inner membrane space between the two. And then we see a bunch of these little pancake looking guys in here. The alkaloids, are the little discs. A bunch of them stacked together, we call rannum, all right? It's a stack of them. All of these guys, they're all green looking because chlorophyll pigments appear green, right? And that's what we're looking at, right? And chlorophyll is going to get excited by the energy from the sun, the photons, and that's going to harness, as we trap this energy, it's going to harness that energy, and we're going to make sugars in this process, right? I'm going to go through all of those details yet. That's chapter 5. We will hit that for sure, but the idea is to use the energy from the sun, photo, to synthesize and produce sugars, right? And that's all happening in the stroma. The fluid, the alkaloids are basically floating in, right? So the photo portion happens here in these discs, and the synthesis happens in the stroma where we make the sugar water, if you will, right? And then from that, we will use that for the plant, right? As it creates that stuff, it then will metabolize through mitochondria, breaking it down and using that to build ATP so that it can do cellular work, so it can grow, and other stuff that plants need to do, right? That's the idea. So we saw this, we saw the membrane, we saw some of the cytoskeleton stuff for the internal structure, right? This is looking at external, on the outside of that cell. There's other stuff. And we showed a few proteins. There's a few details in there, but there's other stuff, these collagen fibers and protein and sugar, fibronectin, that sort of helps to tether and anchor things together. It helps to keep one cell touching another cell and almost sort of like Velcro, linking them up is kind of the idea. So it's extracellular matrix. It's like the internal cytoskeleton, but on the outside, helping to orient in the organism as a whole, not keeping stuff tethered inside, but keeping the respective cells positioned, right? That's kind of the idea with that stuff. And again, it depends on the types of cells, on whatever cell we're talking about as far as what sorts of stuff they have on their surface or not, right? So there are definitely differences dependent upon the types of cells, right? As far as how they come in contact with one another, if they potentially may have the ability to communicate with one another, possibly. All of these things depends on the type of cell. So this very first one up here is 2 plant cells. Both of these plant cells are connected, and technically all of the plant cells exhibit this stuff. So we can look at a gigantic tree, and with all of the connections, technically it's like it's one gigantic cell because they have channels that allow for the movement of stuff, treating it all kind of like it's one unit, right? So this connection in here is called plasmodesmata. These are little pores, little canals from one cell to the next, little channels that allow for the movement of stuff, all right? Down here on this cell, the last bit, are gap junctions. These are similar, but this is an animal instead of a plant cell. These are pores that allow for the same idea, but between animal cells instead of plant cells, right? So the gap junctions and plasmodesmata are pretty close to the same idea, right? It's just different stuff. Over here, these guys are the desmosomes. This is sort of that image I was just showing, and I said it's kind of like Velcro, right? That's what we're really looking at here. So the desmosomes allow for proteins to interact with other cells and tether them together, right? Here is cytoskeleton stuff that we're showing on either side, other proteins that are anchoring, right? But this, linking them together, right? It's not a tight fit. all right? That's the one I haven't talked about yet, all right? But it holds them together, right? The tight fit, these guys are what we call tight junctions, as it sounds. They should seal, right? I like to think of these guys like your shower, right? You want to keep the water in the shower. You don't want it to go out into the wall or out onto the floor. You want it to stay in. And oftentimes they keep that stuff on the inside with some caulking. Caulk the cracks, make sure no water gets through them, right? That's a tight seal, and you want that to happen, right? Same idea with some of our own cells. And we may not want stuff to readily move through, right? So an example of this might be your intestines. You want to keep stuff inside of the intestines. You want to keep that stuff in there, right? You don't want it to just move out, or that would be a pretty poopy mess, right? Not a good idea, right? So it stays in there, and all of these intestinal cells from one cell to the next are full of these tight junctions that fuse them together, right? And so, you know, you've got all of them linked up, holding them together. Maybe this is the outside, so in the abdominal cavity, and this is the inner bit of the intestine, so the lumen, where all of the foodstuffs would be, and other crap as well, right? All within them. And these junctions hold the tight junctions hold a very tight, like welding, not Velcroing these cells to one another, keeping stuff contained, right? So it depends on the specifics, depends on the types of cells, depends on plant or animal, but it depends on the types of cells as far as what sorts of stuff we're seeing, right? And we would not expect to see a bunch of desmosomes or gap junctions in intestinal cells, because that would be bad news, right? That would cause some serious issues. So I mentioned this, and we'll quit right after this, but as we look at this membrane again, it's a fluid mosaic model. And what we mean by that is the potential for it to be pliable, to be flexible, to be able to take on specific shapes, if you will, right? If we're talking bacteria or plant cells, that's not the case, because they have cell walls. They're already dictated, you're going to be the shape. Right? But animal cells, we've got the capability of taking some different sorts of sizes and shapes, dependent upon where we're at. Okay, any questions or concerns? Good. All right, guys, have a great day. I will see you all Thursday.